Hello and welcome to the FH Foundation's webinar on homozygous FH treatments. I'm so pleased to be here with Dr. Mary McGowan, the Chief Medical Officer of the FH Foundation. Hello, Dr. McGowan.
Hello, Kat.
The FH Foundation is a patient-centered nonprofit organization dedicated to research, advocacy, and education for all forms of familial hypercholesterolemia. Our mission is to raise awareness of FH and to save lives by increasing the rate of early diagnosis and encouraging proactive treatment. This webinar is part of our HoFH community programs. And we'd like to thank the sponsors of both those HoFH community programs and also our broader FH community and action programs. We would particularly like to thank the sponsors of our Rare Disease Day campaign, Regeneron, RegenXBio, and Konica. Dr. McGowan, thank you so much for being here and for presenting on HoFH treatments today.
Thank you so much, Kat. It's a pleasure to be here. So I'm sure that most people that are on the line recognize that there are two forms of familial hypercholesterolemia. And our focus today is going to be homozygous familial hypercholesterolemia. But just as a refresher, heterozygous familial hypercholesterolemia is very common, occurring about 1 in 250 people worldwide. These people with heterozygous FH have inherited a single gene for familial hypercholesterolemia. On the contrary, people with homozygous familial hypercholesterolemia have inherited 2 genes for familial hypercholesterolemia. And that leads to very elevated LDL cholesterol and the possibility of cardiovascular disease occurring in childhood. We typically say that people with homozygous FH respond less well to medications than heterozygous FH patients. But one of the great things is that we are continuing to have newer medications and some that people with HoFH respond very well to. As you'll see, as we go through this discussion, there are some medications that are exclusively for people with homozygous familial hypercholesterolemia and others that can be used for both homozygous and heterozygous familial hypercholesterolemia. As you know, homozygous familial hypercholesterolemia is rare, occurring in 1 in 300,000 people. If we could have the next slide. This slide, I would like to focus on the homozygous line. And I would say that, as it says on at the top of this slide, there is an incredible opportunity for prevention. And in the days before we had cholesterol-lowering medicines, the average person with homozygous familial hypercholesterolemia would cross the threshold for coronary heart disease. In other words, would have significant plaque, cholesterol buildup in their coronary arteries by around the age of 12 and a half. We have great reason to hope, and we have evidence right from our homozygous familial hypercholesterolemia community with the FH Foundation. We have homozygous patients that are in their 40s, in their 50s, in their 60s. And I think the future is bright because of the medications on the horizon. Next slide. So what are the available therapies for homozygous familial hypercholesterolemia? There are treatments that we used in the past and then added to over time. So in 1973, the medication that we had available for patients were bile acid sequestrants. And I will talk about those today, but they were insufficient to make a dent in the cholesterol of patients with homozygous familial hypercholesterolemia. 1987 was a watershed year for people with familial hypercholesterolemia. Statins came on the market. We now have 7 statins, but the first statin was lovastatin, and it is a drug that we'll talk about. It's the mainstay of therapy for people with any form of familial hypercholesterolemia. That's the agent we start with. In September of 1997, LDL apheresis, now called lipid apheresis, was introduced. And this is really a sort of a dialysis-like procedure where we, based on the charge of LDL particles, we remove LDL from the bloodstream. And I'll be showing you a schematic of how LDL apheresis works. In October of 2002, ezetimibe came on the market. And ezetimibe, we'll talk about as well, is a cholesterol absorption inhibitor. So it works differently than statins and works in the gut to inhibit the absorption of cholesterol. Lomidopide is an agent specifically approved for patients with homozygous familial hypercholesterolemia, and we'll talk about its mechanism of action as well. One thing to note about lomidopide is it does not depend on LDL receptors to reduce LDL cholesterol. In July and August of 2015, the first two PCSK9 inhibitors came on the market. Evolocumab is specifically approved for homozygous FH, and evolocumab and alirocumab are approved for patients with atherosclerotic vascular disease and heterozygous familial hypercholesterolemia. Just this month, Bempedoic Acid was approved. This is a drug that works in the same pathway as statins, but a little bit north of where statins work. And this is what's called an ATP citrate lyase inhibitor. We will go through these as we go along. And I should mention that just this very week, Bempedoic Acid in combination with ezetimibe has also been approved. Next slide. So I'd be remiss if I didn't talk about diet and exercise and other risk factors. I think it's important, though, for people with homozygous familial hypercholesterolemia to know that it is impossible to lower your LDL sufficiently with just diet and exercise. They are important for many, many reasons, just for a healthy lifestyle, for the prevention of weight gain, but they will not suffice to lower the cholesterol. When we think about diet, we think about reducing saturated fat, calories from fat, and trans fat. We like diets that are high in soluble fiber, and that includes including things like grains and legumes, as well as fruits and vegetables. Some people require referral to a dietitian, and I think that is always worthwhile. We can always learn something from a dietitian. Getting regular exercise is likewise very important. It's important as well for persons with homozygous familial hypercholesterolemia to try to make sure that they don't collect other risk factors, such as high blood pressure or hypertension, diabetes, metabolic syndrome, overweight or obesity, certainly not smoking, and being physically inactive. If you have any of these risk factors, you'll want to work with your healthcare provider to get on either the treatments necessary and make the lifestyle changes necessary to, for example, stop smoking or get physically active. I'm not going to spend a lot of time today talking about lipoprotein a, but lipoprotein a is a genetic cholesterol, and persons with homozygous familial hypercholesterolemia as well as heterozygous familial hypercholesterolemia have an increased risk of having inherited the gene that leads to an elevated lipoprotein a, which is a risk factor for heart disease and stroke. So it's important, if you haven't already had your lipoprotein a measured, to have that measured. Next slide. So as I said earlier, statins are our first-line treatment. They are the mainstay for treatment of homozygous familial hypercholesterolemia. And statins come in 7 varieties, and they also come in intensity, varying intensity, high, medium, and low. And for people with homozygous familial hypercholesterolemia, the goal is to be on high intensity statins. So that would include atorvastatin, 40 or 80 milligrams, or rosuvastatin, 20 or 40 milligrams. So that's the goal. Sometimes people have some difficulty tolerating statins. Most people tolerate them beautifully, but if they do, then using a moderate intensity statin or even a low intensity statin is certainly better than no statin at all. So trying to get on the high intensity statin is most important, but certainly the moderate and low intensity statins are beneficial as well. Next slide. So how do statins work? Statins lower the level of cholesterol in the blood by reducing the production of cholesterol in the liver. Statins block a particular enzyme in the liver, and as you can see, it's this HMG-CoA reductase enzyme. So that's relatively high up in the pathway for the production of cholesterol. Cholesterol gets reduced in the liver cell and other cells, but particularly the liver cells. And as that happens, statins lead to an increase in LDL receptors on the surface of the liver cell, which help remove more LDL cholesterol from the bloodstream and reduce the LDL cholesterol level. Ezetimibe is another cholesterol-lowering agent. It, too, lowers plasma cholesterol, but it does so by a different mechanism. It inhibits the absorption of cholesterol from the small intestine by inhibiting a particular sterol transporter called the NPC1L1 transporter. And by doing so, it decreases the amount of cholesterol available to be delivered to the liver. Less is absorbed from the intestine, so it's excreted in the stool. And when there's less cholesterol delivered to the liver also increases LDL receptors. So much like statins increase LDL receptors, so does ezetimibe, and helps remove LDL cholesterol from the blood. Next slide. So PCSK9 inhibitors. We'll spend a little bit of extra time on the next two slides. These agents came out in 2015, and they have a very interesting mechanism of action. So PCSK9 stands for proprotein convertase subphylicin pepsin type 9. PCSK9 is simply a protein. It's made in the endoplasmic reticulum of our cells. What you're seeing here on this slide is a depiction of a liver cell, but this could be any cell. The liver cells probably make the most PCSK9. So makes PCSK9, and that gets released into the bloodstream. So what you're seeing out there as depicted as blue is the bloodstream. PCSK9 latches on to the LDL receptor complex. And you can see here the LDL has a little yellow ribbon around it. And that yellow ribbon is really something called ApoB. It is the protein on LDL that allows the LDL receptor to recognize LDL. And what happens is the PCSK9 protein latches on to that LDL receptor complex, drags it into the cell, and instead of allowing a separation of the LDL, if we can go back one slide, instead of allowing a separation of the LDL receptor LDL particle, that PCSK9 holds on tight, and the entire complex, the LDL receptor, the LDL, and PCSK9 is delivered to the lysosome where they're all destroyed. So next slide. The LDL receptor isn't able to recycle. The interesting thing about the LDL receptor is the LDL receptor and the LDL, in the absence of PCSK9, separate, and the LDL receptor recycles. An average LDL receptor makes about between 100 and 150 trips in its lifetime, each time bringing in an LDL particle. So we certainly want the LDL and the LDL receptor to separate. We want the LDL to be delivered to the lysosome and the LDL receptor to go up to the surface of the liver cell. So enter the PCSK9s. The PCSK9s are monoclonal antibodies. That's what that MAB stands for. in the left-hand corner of the slide. And what monoclonal antibodies do, in the case of the PCSK9 monoclonal antibodies, alirocumab and evolocumab, they bind PCSK9 in the bloodstream, preventing it from latching on to the LDL receptor complex. So we have more LDL receptors on the surface of the liver cells, and we're better able to remove LDL cholesterol from the bloodstream, and the LDL level goes down. Next slide. Bempedoic acid is, I would say, the new kid on the block, just approved in February of 2020. Bempedoic acid works in the same pathway as statins. It lowers the level of cholesterol in the blood by reducing the production of cholesterol in the liver. And you can see the enzyme that bempedoic acid blocks is something called ATP citrate lyase. This is a couple of steps above where statins work. So they both work in the same pathway, and they both decrease intracellular cholesterol. And ultimately, both bempedoic acid and statins upregulate LDL receptors. In other words, create more LDL receptors to help remove LDL from the bloodstream and lowering the LDL in the blood. Next slide. Bile acid sequestrants are the oldest lipid-lowering medications. They bind bile acids in the intestines, keeping them out of the circulation and depleting the liver of bile. This increases bile synthesis in the liver, which requires cholesterol, and in turn, it also upregulates LDL receptors. So there's a theme here. Many drugs work by upregulating the LDL receptors, although there are some that do not, but upregulate the LDL receptors and remove more LDL cholesterol from the bloodstream. So you can see that here, as an example of a bile acid sequestrant, we have cholestyramine. This is a powdered agent. We also have cholestyramine and cholecibellum, which are tablets that may be a little bit easier to tolerate. The cholestyramine is mixed in water or juice. Next slide. Lipoprotein apheresis is a very important treatment modality for people with homozygous familial hypercholesterolemia. And this is a procedure that occurs either once a week or once every 2 weeks. Blood is withdrawn from one arm and goes through a blood pump and plasma separator. The plasma separator separates red blood cells, and blood cells from plasma. Plasma is what contains the LDL cholesterol. Next slide. And one more slide. Great. And so as we can see here, blood is withdrawn from one arm. It goes through this plasma separator. Then the plasma, which contains the LDL, goes through the plasma pump and finds its way to lipoabsorber columns. These lipoabsorber columns act by charge. So the lipoabsorber columns contain, and I'll be showing you a blow-up of the lipoabsorber column so you'll get an idea of exactly what I mean. They contain cellulose beads, and these cellulose beads are impregnated with dextran sulfate. Dextran sulfate is negatively charged, and the ApoB, which is the protein on the surface of the LDL cholesterol, is positively charged. And so we're able to adsorb the LDL cholesterol. And once the LDL cholesterol fills up the lipoabsorber column, we use a repriming solution to rinse out the column, and we switch over to another column. During this 3-hour procedure, we continuously go back and forth between columns, removing LDL cholesterol from the blood or the plasma, and then the plasma is returned and reunited with the blood cells and returned to the other arm of the patient undergoing LDL or lipoprotein apheresis. Sometimes the blood goes back to a port. People who have this procedure have the procedure on a weekly or biweekly basis and usually require either a port or something called an AV fistula, a fistula that unites an artery in a vein in the arm and allows a large blood vessel for return of blood. Next slide. This is a blow-up of what's in that lipoabsorber column. So cellulose beads that have dextran sulfate that are negatively charged, and they interact with the positively charged LDL to sort of suck up the LDL and remove it from the blood. Next slide. This is an example of what the LDL or lipoprotein apheresis machine looks like. The only company that makes lipoprotein apheresis equipment for the United States is Konica. And this is Tom sitting by a Konica machine getting his lipoprotein apheresis. Tom is an FH advocate for awareness. Next slide. Lomidopide is an agent that is approved for people with homozygous familial hypercholesterolemia only. Lomidopide inhibits something called microsomal triglyceride transfer protein and ultimately decreases lipoprotein production. It should be noted that lomidopide does not depend upon LDL receptors to work. And this is important because some people with homozygous familial hypercholesterolemia are lacking any activity of their LDL receptors. They're called LDL receptor null individuals. So as you can see at the level of the liver, microsomal triglyceride transfer protein unites triglycerides and ApoB to form VLDL. VLDL, or very low density lipoprotein, is the lipoprotein that the liver primarily makes. Once in the blood, that VLDL gets converted to LDL. So by decreasing the VLDL production, we will decrease LDL production. It should be noted that lomidopide should be used in conjunction with a very low-fat diet and other lipid-lowering therapies. Next slide. So I'd like to just talk about new therapies in development for familial hypercholesterolemia now. Next slide. So these therapies are in clinical trials, or they have completed clinical trials and have not yet been approved by the Food and Drug Administration. Inclisiran is a PCSK9 inhibitor, but it's different than alirocumab and evolocumab because it works in the cell, whereas alirocumab and evolocumab work in the bloodstream. Inclisiran's phase 3 clinical trials have been completed in adults, and we are waiting to learn what the FDA's decision is on inclisiran. Evonuncumab is something called an ANGPTL3 inhibitor. It is a drug that lowers LDL. The mechanism of action by which it lowers LDL is not completely understood, but it can profoundly affect LDL in patients with homozygous familial hypercholesterolemia. There is a phase 3 trial in homozygous FH adults that has been completed. And importantly, there is a trial that is recruiting for homozygous familial hypercholesterolemia children, so a pediatric trial. There is still another trial that is recruiting for persistent hypercholesterolemia in adults. So that would mean persistent hypercholesterolemia in the face of other lipid-lowering medicines, maximally tolerated lipid-lowering medicine. ARO, ANGPTL3, or ANG3, is also an ANGPTL3 inhibitor and has an ongoing trial for HoFH individuals. Both alirocumab and evolocumab have trials in pediatric trials. the alirocumab trial is recruiting, and the evolocumab trial is ongoing, meaning that it has fully recruited, and so but is not completed yet. Alirocumab and evolocumab also have a pregnancy exposure registry. So if someone were to become pregnant while on alirocumab and evolocumab, this registry would allow following of both the mother and the child having been exposed to alirocumab or evolocumab during pregnancy. Alirocumab, which was not specifically approved for HoFH yet, has an HoFH trial ongoing. And the HDL acute lipid optimization for HoFH is ongoing. This is testing the effect of infusing plasma enriched in delipidated, so delipidated HDL without lipid in it, with an aim to reduce coronary plaque. The AAV gene therapy for HoFH in adults is a trial that is recruiting. This stands for adeno-associated virus gene therapy. And adeno-associated virus gene therapy aims to fix abnormal genes or to normalize the expression of either overactive or underactive genes. In the case of HoFH, it would be underactive genes. And then finally, there is CRISPR gene therapy on the horizon. There is early stage research underway on a potential gene editing therapy to permanently lower cholesterol aimed at reducing the risk of coronary disease. And so that's what CRISPR is all about. Next slide. So I'll turn back to you, Kat.
All right, thank you, Dr. McGowan. That's really wonderful. I think it's so important for people with homozygous FH to understand what all of their treatment options are. Not every option is right for every person, but at least with this overview that you've given, this really wonderful overview, people will know what all the options are, and they can go and speak to their FH specialist about what is right for them in order to achieve their LDL lowering goals. What we'd like to share with you is also what the FH Foundation has to offer for individuals with FH. So one of our resources is an interactive map on our website where you can enter your zip code and your address and find an FH specialist. And we have over 350 specialists listed in the map from all over the world. You can find it on the website at fhfoundation.org/find FH specialist. If you have trouble finding someone near you, please feel free to contact us at the FH Foundation and we'll help you find somebody. When you visit the FH Foundation webpage under this topic of familial hypercholesterolemia, you will find this webpage specific to homozygous FH, and you'll find more information there, videos, and links to additional resources specific to homozygous FH. Again, you can find that on the fhfoundation.org/whatisHoFH. We'd like to let everybody know that we have a program where we offer no-cost genetic testing from the FH Foundation in partnership with Invitae and Genome Medical for people who have homozygous FH or may suspect that they have homozygous FH and are looking to clarify their diagnosis. Genetic testing is, again, another important decision to make, and it's best to have genetic counseling before you make the decision to go ahead with genetic counseling, and this offer includes pre-testing genetic counseling. If you'd like more information about this offer, please email us at info@thefhfoundation.org and put HoFH community in the subject line. We have a place on our website where you can look for more information on clinical trials. There are always new clinical trials coming on, clinical trials that fill their recruitment, so they're no longer recruiting, and then others that open their recruitment up. So things are always changing when it comes to clinical trials, so it's important to visit the page and see what's new and what you might want to consider. I think it's really great to find out what what you might want to join when it comes to a clinical trial. Again, another important discussion to be having with your healthcare provider and with your family, but also just to know what is potentially on the horizon for treatment for homozygous FH. So again, that's on our website, looking under research and clinical trials. Please sign up for our newsletter to stay informed and to get news of other webinars such as this and activities that we have. We have in-person events, we have online events, and we are always trying to get important information out to you. So you can join us by entering your email on our website. And we'd like to thank you. If you have any questions, you can always email us at info@thefhfoundation.org. Thank you, Dr. McGowan, for this great presentation, and thanks everybody. This is Kat Davis Ahmed from the FH Foundation. It's been wonderful to have you with us.
And thank you, Kat. This was a pleasure to participate in, and I look forward to doing more.
Thanks.